Proteins. So the basic unit of a protein is your amino acid. Within the chemical composition of a protein, there will always be carbon, hydrogen, oxygen, and nitrogen. Sometimes there's sulfur and phosphorus. Your main source of protein could be meat. Your structure is folded, okay? So protein is folded. So it's amino acids joined together to form a chain, and that chain is a protein. So there's 20 different types of amino acids found in a protein. A peptide bond forms between the amino acids. Polypeptides consist of more than 20 amino acids, and proteins are very long polypeptide chains, and they will consist of more than 200 amino acids. Now here we have an amino acid forming a polypeptide chain which forms a protein. And you can see here we have our peptide bond between your amino acids. So the number and sequence of the amino acids in a chain determines what type of protein it's going to be. So the way the chain folds gives the protein its qualities. Now, of the two types of proteins, we have fibrous and we have globular. So a fibrous protein has a few or no foldings. Our example of a fibrous protein, we need to know one, I'm providing you with two. Myosin. So myosin, we would find myosin in muscle. The other example is keratin. It's found in hair and nails. A globular protein, so a globular protein here has many foldings. Your main example is enzymes, and enzymes are catalysts. Another example of globular proteins are hormones. These are chemical messengers. So your function, the structural role and metabolic role of proteins. So your structural role, we're using our examples from our fibrous proteins. So you could say keratin found in hair and nails, or myosin found in muscles. Your metabolic role is to do with your globular proteins. So your metabolic role, we can say hormones are proteins, or we can say enzymes are proteins. And this leads us to the mandatory experiment for protein. So the chemical that we're using here, the reagent that we're using here is burette. You could say sodium hydroxide and copper sulfate if you wish, but burette is accepted. So into a test tube, I'm going to add a source of protein. So I would put milk into the test tube, I'd add my burette, shake it a little bit, and a positive test for protein, it'll go from blue to purple, lilac, mauve. Your control on this would be water. So into a test tube, I would add water, I would add burette, give it a little shake, and it'll stay blue. So a negative test for your control, water will stay blue.